How's it going, everyone? It's Chow here, and today we are going to discuss a little bit about transcription, so let's get started. Okay, so transcription is kind of a tricky topic, and I'm not exactly sure 100% what's the best way to teach it, so I'm going to go through some of the basics, and hopefully you will have a better understanding of transcription after this video. So transcription is very critical in the body because it is actually what's going to be turning a DNA sequence that codes for some kind of a gene product, and it's going to turn that DNA sequence into an RNA sequence. That RNA sequence is then going to go into translation, which is another thing that happens after transcription, where you have the RNA strand that's going to be used as a template and read and eventually translated into an amino acid sequence. And this amino acid polypeptide can be turned into a gene product or some kind of a protein. So the starting step is with transcription, again, turning that DNA sequence into a complementary RNA sequence. So very important first step. But obviously, if you're talking about transcription, we have to discuss the central dogma. So the central dogma is this straightforward pathway where you're taking DNA through transcription to make RNA, and then you use that RNA through translation to make a polypeptide, which can potentially be something like a protein. So DNA, transcription, mRNA, translation, and then protein or some other gene product. So this whole concept is called gene expression. That's another term for it. And then when we talk about controlling gene expression or controlling parts of the central dogma, we're talking about regulation of that gene expression. Are you going to increase it? Are you going to decrease it? Are you going to repress it altogether? This all falls into the category of control. So when you look at transcription, one of the things that is very critical is that it occurs in the nucleus with this enzyme called RNAP or RNA polymerase. Afterwards, that mRNA passes through the nuclear pore in the nuclear envelope, and then it's going to be translated by the ribosomes in the cytoplasm. So obviously, this is something that is going to be happening in a eukaryotic cell because we have a nucleus present. In a prokaryote, it's going to be a little bit different because you obviously don't have this nucleus, so you can actually have transcription and translation happening at the same time. But with eukaryotes, it's going to be a little bit more compartmentalized. So again, prokaryotic cells can have transcription and translation happening at once because you don't have that nuclear barrier, you don't have that nucleus. But when you have eukaryotes, you have a little bit of compartmentalization, and that means that you have transcription that's happening specifically in the nucleus, and then translation that's going to be happening in the cytoplasm with those ribosomes. So there you go. Now, one of the key enzymes of transcription is the RNA polymerase, or RNAP as it's sometimes written. There are actually a couple of different kinds. So each different type of RNA polymerase basically transcribes a different type of RNA. So we have RNA polymerase 1 that transcribes largely rRNA, and basically I think it's all rRNA, whereas we have RNA polymerase 2 that's going to be transcribing our mRNA or messenger RNA, which is eventually going to be read into a protein. And then we also have RNA polymerase 3, which is used to transcribe some small RNAs as well as your tRNAs, so your transfer RNAs. So for transcription, we're really going to be talking mostly about RNA polymerase 2 because we're going to be turning and reading that DNA strand to make an mRNA strand, which is eventually going to be read to make a protein or some kind of a gene product polypeptide. So there you go. Now, when you look at RNA polymerase, it actually shares many type of properties with DNA polymerase, that enzyme that we use to replicate DNA. But what's really different about RNA polymerase is that it doesn't require a primer. So if you think back to our whole discussion of DNA replication, remember that DNA polymerase requires a primer to basically go on and actually replicate the strands. RNA polymerase can actually catalyze a lot of these reactions without the primer. So it can basically put on RNA bases without the need of a primer. However, the reaction itself about putting on and extending and elongating that mRNA strand is going to be the same as basically with DNA polymerase. It's just making it longer and adding more bases onto the strand as time goes on. And just like DNA polymerase, it's going to be synthesizing that new strand in the 5' to 3' direction. Of course, one of the things that we should look at when we talk about RNA bases, again, is this 2' carbon here. It actually has an OH group, so a hydroxyl group, whereas in DNA, those bases will have basically just a hydrogen. So this is an OH group, which means this is basically an RNA base, whereas if this is a hydrogen, it would be a DNA. So this is a key difference between the bases themselves. So that's really nice and all, but if we look at the RNA polymerase itself, we also realize that just like DNA polymerase, it's going to be reading, right, that template strand from the 3' to 5' direction. So this blue strand down here is the template strand. It's the DNA template strand. And this I guess a pinkish, orangish blob over here is supposed to represent your RNA polymerase. And the RNA polymerase is going from left to right, which means that if you look at the reading section of it, which means where is it reading on the DNA, you can see that it's reading this bottom strand, the template strand, from the 3' to 5' direction. So one thing to keep in mind is that DNA polymerases and RNA polymerases always read the template strand of DNA from the 3' to 5' direction. But when it comes to synthesis, right, the synthesis of this, in this case an RNA strand, it's only going to be synthesized in the 5' to 3' direction. So one thing, again, to keep in mind is synthesis is 5' to 3', but reading is 3' to 5'. Okay, that's a lot. So if we want to think a little bit more about the polymerase itself, you can go ahead and maybe look at how it potentially differs from the DNA polymerase and also think about how it's similar. So that might be helpful if you want to get a better understanding of transcription. Now, when you make that RNA strand, there are actually a few different types of RNA available to be made, again, depending on what type of RNA polymerase you're actually using to transcribe the DNA. So we have one of the most important ones and well-known ones, and that's mRNA or messenger RNA. This is the type of RNA that basically carries the gene sequence, which eventually can be made into some kind of a polypeptide or a protein. There's also rRNA, which is ribosomal RNA, and that's going to be found in the ribosome, and it can be used to help kind of catalyze peptide formation during translation. We also have snRNA, these small nuclear RNAs. They are generally used during RNA processing, as well as these non-coding RNAs, and they are often used for regulatory components. There are also other types like siRNAs and microRNAs that have other functions as well, but probably the most important ones to keep in mind are the first two over here. So when you look at the transcription process itself, what's really fascinating is that it's kind of broken down into three major steps. We have initiation, elongation, and termination. So with initiation, basically, RNA polymerase comes in and finds the start site on the DNA. Elongation is basically when RNA polymerase leaves the start site and starts synthesizing that RNA. That's when it's like rapidly replicating the RNA and making more of that strand. So that's basically elongation. I guess a better way is not really replicating, that probably is a bad thing to say, but it's adding on more bases onto the ever-growing RNA strand. So that might be a better way to put it. So don't think of it as replicating. And then finally we have termination. And termination is basically when it just kind of finds a stop site and it just stops. And the RNA strand is finished. So that's basically the rundown of initiation, elongation, and termination processes within transcription. So if you look more specifically at initiation, One thing that's very critical is how in the world does that RNA polymerase find where to start? So transcription initiation only begins at specific sites, and at least in a lot of eukaryotes, it's aided by proteins called transcription factors, or also known as TFs, in eukaryotes. So they can act as switches to basically turn on or turn off certain amounts of certain genes or certain amounts in correlation with other things like enhancers and repressors. And basically with initiation, it begins at a transcription start site, or TSS, where that RNA polymerase binds. Now, when you look at something like a prokaryote, it's slightly different. Because prokaryotes, the RNA polymerase has sort of a sigma subunit that kind of binds to the specific start site sequences within the promoter region. So there's one at the -35 region and one at the -10 region, and that's how basically the RNA polymerase is able to recognize the initial sites of that particular gene that needs to be transcribed in prokaryotes. So keep in mind that the binding is over here in -35 and -10, but really the transcription start site is down over here. So recognition is up here, but the actual start site is slightly downstream. So that's one thing to keep in mind. Now, when you look at initiation in eukaryotes, they actually are a little bit different because the RNA polymerase lacks a subunit that recognizes the specific start site sequences. So instead, we have an RNA polymerase that actually interacts with other kinds of factors called general transcription factors to basically help find the start sites. In particular, we have TF2B and TBP, which can help separate strands. So there are some very important proteins that can come in and basically help out with that process because there's really no subunit that recognizes a specific start site. So there you go. Now, with transcription, you have to regulate it. So there are kind of a couple of questions you can ask. And the first one is where? Well, transcription only begins at specific sites, so that's basically the where question. The when question can be answered by looking at how transcription initiation is controlled by certain regulatory transcription factors, such as activators and repressors. And then obviously how much, which is the level of transcription, and how much is going to be going through that whole process of transcription, how much protein is going to be made at the very end. And this is often going to be looking at affinity of the RNA polymerase to specific sites, as well as transcription factor affinity, which bind near the RNA polymerase. So these are some ways you can try to question regulation of transcription. So, when exactly does transcription occur? Well, oftentimes transcription occurs through regulation via these things called activators. So activators are things that kind of recruit RNA polymerase and activate transcriptions. On the other hand, you have these things called repressors, and repressors do the opposite of what activators do, and that is they basically bind and prevent transcription from happening. There are also other types of sort of repression and activation, and one is with the lactose operon. So this is actually one of the situations where you actually need something, the presence of something, in order to start transcription. So normally in the cell, you have a repressor that binds to this operator region, and it prevents RNA polymerase from transcribing genes. However, with lactose, the lactose actually binds to the repressor, changes its shape, and now the repressor is no longer able to bind to the operator region, and so now the RNA polymerase can go ahead and transcribe those genes to basically make proteins which eventually will, for instance, degrade that lactose. So that sort of allosteric regulation of this repressor by lactose is able to help control whether the cell is going to make a bunch of enzymes to break down lactose in the presence of high amounts of lactose or not make it at all because there's no need to if there's not any lactose around. If you want to look kind of at the grand scheme of things with these concepts of promoters, enhancers, genes, etc., you can basically look at a diagram like this, where you have this beige colored thing, which is the RNA polymerase. You have this, I guess it's oh, these two are kind of similar in color, but this bluish teal colored thing over here is the gene that's going to get transcribed. The yellow here is the promoter. We have the purple here, which is basically a general transcription factor. We have the red, which is a more specific transcription factor. And then we have this over here, which can be an enhancer or potentially maybe even a repressor. But in this case, we're going to talk about an enhancer. And this loop that comes back, so the enhancer is actually going to be upstream or downstream of the actual transcription start site. But by regulation through these intermediate proteins, transcription factors, etc., sometimes you actually have like mediators in there, and these things all together can help kind of regulate the amount of a protein that's produced. So without the enhancer, sometimes you can still produce the protein product, but without the enhancer, it produces less. If you actually connect it to the enhancer and let that enhancer signal everything over here, it can actually make more of that gene product. So depending on the basically the presence or absence of enhancers, you can make more or less of a gene product. So this is a very critical step in transcription, because transcription is not necessarily just about making that protein, making that mRNA strand, which can be made into a protein, but it's also about regulation. How much of that mRNA are you going to be producing? How much of that protein are you going to be making in the very end? And there are lots of ways to regulate the process. So that's about it. That's all I got for transcription. I hope you all found it mildly useful. One thing that can be useful also is if you go online and check out an animation. I think that's a very critical step in learning, especially if you're a visual learner. And since that's all I got, I hope you all enjoyed the video, and as always, best of luck studying, and I will see you in the next video.